I'm looking forward to hearing this talk.
Oops.
So, I'm going to say good afternoon, everyone, and good afternoon again. And it's with great pleasure I welcome you here on Earth Day, big 51 birthday, and I'm going to give you a little bit of background to how Earth Day began. Senator Gaylord Nelson, who was a junior senator from Wisconsin, was going to give some talks, teach-ins, as we used to have them back in those days. And Dennis Hayes was a 25-year-old, maybe a couple years older now, student at Harvard. And when he heard that there was going to be an environmental teach-in, he went down to Washington hoping to convince Senator Nelson to let him organize a teach-in at Harvard and maybe other colleges in and around Boston. And 2 days later, as a result of that, Hayes dropped out of the John F. Kennedy School for Government to coordinate a national event, Earth Day. And interestingly enough, he's a Washingtonian and he's from Camas, Washington. So our efforts in Washington to be involved with Earth Day and conservation is not new. It's been going on for longer than 51 years. And our indigenous groups have been fighting for conservation for many, many years before that and continue to. The first day, April 22nd, 1970, climate change, deforestation, and chemical intensive agriculture had yet to become existing crises, if you can believe it or not. And he organized this massive event and then went on to organize over the next 50 years, the Earth Day Network, which extends to many countries. So Earth Day has been recognized as the beginning of the modern environmental movement and is now an international holiday. So from 2004, Washington Native Plant Society started working on Native Plant Appreciation Week, in which we had hikes and lectures and plant sales, and this grew and developed. And then in 2019, WNPS board approved making it a month-long event. And our first event was supposed to be April 2020, and I don't have to tell you all, the COVID hit. And due to the quick action of Denise and others, we pivoted to being an online event. And this year we had 28 webinars, 24 of which were free, scheduled, and they are recorded and you can go back and view them all the way back to last April. So any webinars that were recorded over the past year have been saved and they're there for you to see. Something really special happened in March. The United States Senate voted on a bipartisan proposal, and they voted unanimously. It was to name April 2021 National Native Plant Month. And that resolution was introduced, co-sponsored by Senator Hartman in Ohio and Senator Hirona in Hawaii, Mazie Hirona. And for our United States Senate to vote unanimously on anything was pretty amazing. So here we have it. Today is Earth Day, National Native Plant Month, and we're part of the Washington State Native Plant Appreciation Month, in which we want to say that we would like folks to educate, appreciate, and advocate for our native plants. And in the wonderful proclamation signed by our governor, Jay Inslee, he writes that there are over 3,000 native plant species in Washington State, over 350 of which are listed as rare by the Washington Natural Heritage Program, and how important our native plants are to protecting our natural watersheds, recharging natural aquifers, modulating stream flows, filtering water, reducing erosion and flooding, and protecting our salmon, a wonderful resource. So I'm now going to introduce Sarah Hamman. Sarah is the Director of Science for the Eco Studies Institute, a conservation nonprofit, based in Washington State. Her work is aimed at researching and restoring rare plant species habitat in Pacific Northwest prairies and oak woodlands, using rigorous science and collaborative conservation principles. She holds a BA in biology from Wittenberg University and a Ph.D. in ecology from Colorado State University. Sarah is also an adjunct professor at the Evergreen State College, where she teaches restoration ecology, fire ecology, and soil ecology in the Master of Environmental Studies program. She is vice president of the board for the Olympia Coalition for Ecosystems Preservation, a nonprofit land trust focused on habitat conservation in Olympia, Washington, and the chair of the Research and Plant Inventory Committee for the Washington Native Plant Society. When she's not working, which doesn't sound like she has much time, Sarah enjoys baking, knitting, and adventuring in the mountains, learning all the new plants and mushrooms along the way. Today's talk, she describes as 1 teaspoon of soil hosts up to 1 billion bacteria, meters of fungal hyphae, thousands of protozoa, and tens of nematodes and arthropods. These organisms are constantly making war and peace to manage the nutrients, water, soil structure, disease organisms, and plant roots that make all other life on Earth possible. As we strive to clean our water and air, restore wildlife habitat, sequester carbon, and remediate degraded lands, we must understand the role that soil ecology plays in the success of these restoration efforts. Sarah will discuss ways that soil's knowledge can be used to improve the success of restoration of native plant communities here in our beautiful Washington state. Thank you, Sarah.
Thank you, Gail, and thanks for providing that full intro. Really appreciate it. And thank you all for being here on, again, a beautiful spring day in front of the screens to learn about soils. It touches my heart that there's people that are that are interested in learning about this. So yeah, today I'm going to talk kind of give a broad sweep about a lot of what we've come to learn, we being kind of a larger we of the research and restoration community, around soils and how soils can be incorporated into, soils knowledge can be incorporated into our restoration efforts. When we think about restoration, most of our restoration goals and management goals really revolve around increasing habitat quality and ecosystem resiliency. And we have developed kind of a series of methods or strategies to get there. Usually we start with a degraded site and through a series of site preparation steps, usually including invasive or non-native species removal, native seeding, or establishment of native plants, and potentially reintroduction of rare or endangered species, we end up with what is, at least temporarily, a restored site. Often there needs to be recurring management to maintain that restored system. But when we think about this, so often we think about all of those organisms that we can see above ground. We can see the plants, we can see the insects, we can see the mammals, and we're thinking about the trophic structure or the soil or the food webs that exist above ground. But I would argue that we need to be thinking just as much about the food webs that exist below ground and how the organisms and the incredible complexity that exists below ground can work to support the goals that we have for some of the rare species and habitats throughout our state. So the soil food web is complex for sure. This is showing, I think, at least five trophic levels in the food web, ranging from the soil, the roots and the plants that are in the soil, and all the way to some of the small mammals and even birds that utilize, either live in or utilize the soil system. And I'm actually going to talk about a lot of these and the different kind of trophic levels, so I'm not going to go into it all right now. But this just kind of shows a little bit of the complexity of how these organisms interact with each other and interact specifically with the plants that we're focusing on with a lot of our restoration work. So there are several aspects of the soil system that are extremely important in terms of creating and maintaining this complexity for all the organisms that are there. These include things like soil structure, soil texture and type, soil nutrients, and soil biota. And so I'm going to go through each one of these and talk about how what we know, what we've learned, just some cool factoids about each of these, and also what kind of strategies might be used in each scenario to both inform our restoration and potentially improve the work that we're doing. So when we start thinking about soil structure, the first thing I want to point out is the horizonation, it's called. So the formation of different horizons within the soil. So here on the left, you can see that basically how soil is formed. It's a very long process in most systems that really initiates from weathering. So rain enters the ground, plant debris accumulates. As this says here, the worms churn, the microbes and the fungi metabolize. The roots weather some of the materials. There's downward percolation of water and nutrients, and then accumulation down in deeper layers of the soil of some of those nutrients and ions and metals and things like that. And so the horizonation really ends up for most soil types with these layers. The O layer, which is really the organic layer, and that's where a lot of the soil activity is happening. That's where the O and the A layer, the topsoil, is where most of the roots are. Then you get into the transition zone, the E layer. That's where a lot of leaching takes place. So a lot of water and nutrients kind of move through that transition layer and settle out into the subsoil. And so you can see, depending on how deep the roots are in the system that you're working in, or in the site that you're familiar with, that's going to impact where the nutrients are stored, how deep the plants are able to access different resources. And again, the depth of these layers really depend on the ecosystem type and the climate. So, here this shows them almost about the same depth. That's going to vary, the A versus E versus B versus C. Those layers vary quite a bit depending on where you are in the world. And I'm going to just kind of leave the general topic of horizons there, but just so you know, if I talk about the O horizon or the A horizon, or think about rooting systems, it kind of helps to have this picture in mind. So also when you're thinking about soil type and soil structure, it's helpful to think about soil taxonomy. So for most of us, we are pretty familiar, or at least somewhat familiar, with plant taxonomy, and especially how some of the how it can get challenging when we learn new information and species names change because things are reorganized based on new information, particularly with DNA work that molecular work that we've been able to use over the last few years. Well, there has been significant work throughout the world to characterize the soils of the world. And basically, there's a similar structure in the taxonomy, starting with order and going all the way down. Instead of to species, it's going to series. And so now there are 12 soil orders identified throughout the world. And these are really characterized by the regions and the plant communities that grow on them. And so you can see here, they're organized, or they're listed here on the right with kind of general characteristics of each of the orders and the area globally that they cover. And so gellisols, for instance, are really in kind of higher alpine and higher latitude areas. Spodosols are in forested sites. We have a lot of andosols here in the Northwest because they're formed on volcanic parent materials. And then iridosols in the desert regions. There's mollisols are typical of grasslands. And I think here in Washington, I'll show you, I have a map here. We have, I don't have a map just of Washington, but here of the US, you can see the range of soil types that exist, or soil orders that exist throughout the country. Again, these are just the 12 soil orders. Here, I think it's actually showing 13 because it's showing this gellisol and septisol mix. But then when you get down into within each order, there are thousands of different series. So I think we have at least 8 or 9 of the different soil orders just here in Washington. You can imagine, because of the topographic variation and the climate variability from east to west, we have, and the influence of our volcanoes, really unique and interesting soil profiles throughout the state. So yeah, we have one of the more diverse states when it comes to soil types. And if you're interested in learning more about the soil type that you have on your property or on a work site or just in the region, there's a great resource here that you can utilize to learn more about the local soil, or wherever you want to look in the US. And this here, if you haven't heard of it, it's called the Web Soil Survey. This is through USDA, Natural Resources Conservation Service. And basically what you need to, you have the website here, so you can look at that if you want to come back and look at these slides when they are posted. You can click on the Start WSS, Start Web Soil Survey. And that will bring you to a screen where you can basically start mapping and identify the area that you want to work in. So here you're going to identify your area of interest, and you do that by, you can just barely see this little AOI button. There's 2 AOI buttons, area of interest. You can either draw a square around it or draw a polygon around an area that you're interested in. And I'm here in West Olympia, so I just drew a little square around West Olympia and took some screenshots here. And I know this is hard to see, but basically all these little lines on the map are showing the distribution of the different soil types. You can barely see, there's different numbers here, 52, 51, I can't even see that, 102. And so you can see there's a wide range of different soil types. And if you look on the left, they are listed out on the left. And so you can then look at all the different soil types here identified as map unit name. Click on that and it will take you to another kind of pop-up page giving you information about that soil. And you can look at the Soil Data Explorer and actually download the soils data for your area of interest. So it's just a great resource to get information about the local soils information if you want to learn more about where you live or where you work or where you are, or anywhere that, within the US. One thing to point out here, you can see this is talking about gravelly sandy loam. And alderwood is the soil series. And if you click on this, I didn't take a screenshot, but it shows more information about the soil taxonomy there. But the gravelly sandy loam gives you a little bit of information about the soil texture. So soil texture is the next kind of piece of information that is really helpful to have when you are thinking about how well plants might get established at a site, how well nutrients might be held in a soil if you're going to be fertilizing or using reverse fertilization by removing nutrients. There are, considerations of the soil texture that can influence some of those decisions. So here, this shows the three different types, the three different soil textures: clay, silt, and sand. And usually when you're trying to figure out the type of soil that you have, if you, for instance, send your soil away to a laboratory, they will actually calculate by doing different float tests of the soil, calculate how much you have by weight or by volume of these different textures. You can also do this, though, in a more qualitative way at home, or just wherever you are, wherever you want to test the soil. And actually, before I go into that, I just want to give a reference image here. So this is showing the relative sizes of the sand, which is this larger structure, the silt, which is this tiny one, and then the clay, which is just the little point in the middle. So you can see clay particles are substantially smaller, thousands of times smaller than a sand particle. Therefore, there is larger surface to volume ratio for clay particles, right? So clay particles can actually hold more nutrients per unit volume than a sand particle can. And so if you have extremely sandy soil, there are things that you can do to increase the clay content to potentially be able to hold on to nutrients better, or to retain nutrients in that soil system. Sand has, because of the large particle size, sand has really high leaching and drainage capacity. Not surprising, right? But that's something to consider if you are going to be trying to irrigate or do any kind of water additions for supporting plant establishment, for instance, on a site. If you have really sandy soils, a lot of that is just going to leach out and carry a lot of the nutrients with it. So it's helpful to consider the proportion of sand to silt to clay. And I apologize for the small text here. I don't expect anyone to be able to read this. I just wanted to show the structure of this kind of step-by-step test. So the USDA NRCS, the Natural Resources Conservation Service, has kind of a step-by-step soil texture hand test that you can use. And if you just type in Google, soil texture test, it should come up. And basically you start with some soil and you wet the soil and you go through a series of steps to determine how gritty it is, how sandy it is, how well, if you roll it up, if it can form a ribbon, and how long that ribbon can extend before it breaks. There's a couple set of questions that you follow, and it will, as you go through the process, can determine if you have sandy loam, silty clay loam, those combinations. So that can give you more of a qualitative assessment of what type of soil texture you have. Okay, now I'm gonna shift gears a little bit and shift into some of the slightly more complex nature of soil functioning, structure and functioning, and start with the carbon cycle. So as most of us know, I'm sure we all know, we are thinking a lot more and concerned a lot more about carbon and carbon sequestration and carbon emissions now than we ever have been in the past, right? And here, this image here is just showing in a very simplified way the carbon cycle that shows the pools, the atmospheric pool, the vegetation pool, the sediment pool, the ocean pool, all the pools of carbon and the estimated gigatons of carbon that exist in those pools. So the atmosphere holds about 750 gigatons of carbon, for instance. This also shows the fluxes in purple arrows, and there's purple numbers next to those fluxes going from and to the different pools. So here again, you can see the atmospheric pool holds about 750 gigatons of carbon. The vegetation pool holds 610 gigatons of carbon. And the soils pool here holds 1,580 gigatons of carbon, more than all the vegetation on the planet, more than what's in the atmosphere. The only thing that's substantially larger is the deep, and it is substantially larger, is the deep ocean pool, which holds 38,000 gigatons of carbon. And this here is showing the fluxes of fossil fuels into the atmosphere, which is obviously of great concern. So here, because soil has such a large carbon storage capacity, it has been recognized as one of the areas that we might be able to actually work on to increase the carbon sequestration. And I will say that, obviously the fluxes from things like agriculture and deforestation and fossil fuel burning are all things that we have to be considering and changing the way we manage those lands and those resources. But there are some things we can do with managing our soils that can help to increase sequestration. And so that is more and more becoming a part of restoration goals for different projects, to actually have soil carbon sequestration as one of many restoration goals. And this really kind of sums it up here with this quote by a scientist that's working with the World Economic Forum, is that one of the three great biospheres, soil, ocean, and atmosphere, we have the best chance of fixing soil in the near term. And really that's through changes to our agricultural practices and prioritizing this idea of moving carbon below ground to safe places. I'm going to talk a little bit more about what that means. But here on this map, this is again from the World Economic Forum, they've just identified places throughout the world that have the greatest potential to increase soil carbon stocks. And that's largely through changes in agricultural management and reducing deforestation and replanting trees, and really kind of rethinking how we manage our soils. So this is an image that came from the Sage Grouse Initiative here in the interior West, but Eastern Washington and Eastern Oregon. That I just love this image showing the relationship between the above ground and below ground parts of the plants that we know and love. So not just conserve our native plants, but conserve the roots of those plant species. It's easy to think about old growth forests and the just jaw-dropping beauty and greatness of huge old growth trees. It's a little bit harder to think about old growth grasslands and prairies and shrublands when the majority of the biomass, the majority of that greatness is below ground, and the majority of the complexity there is also below ground. But this image provides a little bit of a glimpse of that, with the sagebrush roots extending down to 6+ feet, and lupin roots also extending down 5 or 6 feet. And then this also shows, I think this is an image of cheatgrass, a little annual weed, and the piddly little root systems that they develop. And the threat that generates to a lot of the arid lands in the West, where you have vast acreages covered with invasive annual grasses that are basically not putting anything in terms of their resources below ground. So it's taking it's removing that carbon storage capacity from those sites by having the invasive weeds, annual weeds present. So these are guidance, areas of guidance that have been developed for agriculture, but I think they apply equally to restoration situations. Here you can see this on the left, it's showing some different management practices that have been recommended to increase carbon inputs to the soil and reduce carbon losses. And these include things like having cover crops and increasing crop residues, basically limiting or restricting the amount of time that soil is sitting bare without plants present, converting as much as possible to perennial systems with grasses and legumes that have those deep rooted systems, using no-till or conservation tillage, and then utilizing conservation grazing strategies, or strategies that are going to be promoting regrowth and support the long-term sustainability of the perennial species on the sites, and ideally bringing in a diversity of native plant species as well into rotational grazing systems that can promote a variety of ecosystem services. So on the right, you can see this comparison of annual wheat to perennial wheatgrass at different months of the year. With September, there's basically nothing present for the annual wheat, but there's this long extended, 2 meter plus 3 meters more like root system. And then in December and March and June, the annual grass is growing, but still rarely gets past about 1 meter. And the whole time you have this extensive root system that's supporting a diverse below ground community throughout the year. So having that below ground structure from the native plant roots really helps to maintain the soil organisms and the soil nutrient cycling that is necessary for the above and below ground systems to persist. So I'm going to jump a little bit into the soil nutrient availability and soil nutrient cycling, and talk a bit about why this is important for restoration as well. So I know there's a lot going on in this image. Really just want to kind of highlight some of the pools of nitrogen that exist. The first being the atmospheric pool. And this is nitrogen in the atmosphere is not available to plants, right? Generally, it's dinitrogen gas, has a really strong bond, almost impossible to break. The only things that can break that bond are lightning, legumes, or basically the bacteria that live in the roots of legume plants, and the Haber-Bosch process that we have to use a lot of fossil fuels for to create, that humans have developed to create fertilizer. And so anyway, this nitrogen does find its way from the atmosphere into the soil, whether it's through fertilization of crops, whether it's through legumes and the bacteria that live in their roots, or whether it's through lightning strikes. And once that nitrogen is in the soil and cycling and inorganic matter, it can be decomposed by a variety of different organisms into inorganic forms like nitrate, nitrite, ammonium. These types of nitrogen, the inorganic forms of nitrogen, are then available for plant uptake. So understanding the availability of the inorganic forms of nitrogen can be helpful to understand if you have a site that is over fertilized, that might provide basically too many resources and give a competitive advantage at times to non-native weeds. If you have a lot of nitrogen on the site, often you're going to be battling weeds for a long time because they are able to grow faster and capitalize on those nutrients often more quickly than the native plants can. So knowing a little bit about the nitrogen content of the soils and the availability of that inorganic nitrogen can be important in understanding how to manage invasive weeds on your site. This is often a problem we have in Western Washington prairies, where we have one of these plants that I'm sure everyone here recognizes, Scotch broom. It's a non-native legume. And so just like the native legumes, it's able to take that nitrogen from the atmosphere and convert it through the organisms in its roots to an available or inorganic form of nitrogen and use that for growth. But it's able to really take advantage and grow quickly in these low nutrient systems because it's creating all of its own resources, basically. So once we remove Scotch broom, there's still often kind of a long-term legacy for a lot of reasons, but sometimes, in many cases, because of the elevated soil nitrogen content. The legacy can also persist of Scotch broom due to toxic compounds, allelopathic compounds they're called, that they can emit into the soil. I think there's 14 different kinds of allelochemicals that can be exuded or emitted into the soil through the plant roots and the stems that can really inhibit growth of native plants. Scotch broom also has a unique mycorrhizal, a fungal community associated with its roots that can be that can inhibit the growth of other mycorrhizal species and other native plants. And as many of you know, its seeds can last for decades in the soil. And so it has all of these mechanisms below ground that can help itself persist, but keep other plants from getting established. So one way that if you do have a species, a legume, a non-native legume, or if there's been a history of fertilization on the site from agriculture, or if there's high nitrogen deposition from some industrial area or an urban area, there are ways to temporarily reduce the available nitrogen in the soil to help get native plants established. Since native plants are going to grow a little bit slower and their phenology is a little bit different, if you can reduce the amount of nitrogen that's available for a period of time when the native plants are getting established, that can help them resist invasion of other non-native species that would otherwise take over. So one approach that can be used is to add carbon to a site. So this could be in the form of sucrose, like literally table sugar, or sawdust, which is typically quite a bit cheaper, but they have slightly different effects. But they can be used by adding these to the soil. There's been a lot of research done around the world on these methods to temporarily reduce nitrogen. Basically, the carbon acts as food for the bacteria and the fungi in the soil. And if you add that food, they're going to start eating the carbon and reproducing. And in the process of growing their populations, and expanding, they will be kind of gobbling up all of that inorganic nitrogen that's in the soil, all of that ammonium and nitrate, and locking it into microbial cells. So they use it for proteins and amino acids and things like that in a way that's no longer available to the plants. So by what's called microbial immobilization, it's actually locking the nitrogen into the microbial cells for a certain period of time where native plants can get established and the non-native plants are not able to basically capitalize on the nitrogen in the system. So that's one strategy that's been shown to be fairly effective. Most of the work has been in grasslands, and that's where a lot of these invasive species challenges exist with nitrogen. So getting into phosphorus, this is another one of the primary nutrients that we have to consider with our restoration efforts. So phosphorus availability really changes and is linked relative to other compounds like iron, aluminum, and calcium. And those all change relative to pH. And particularly the way carbon interacts with those different compounds responds to pH. So here, if you look at this graph, if you start at pH 3 on the left and go all the way up to pH 9, so again, pH 3 is very acidic, pH 9 is very basic, very alkaline. Usually most plants like it, pH 5, pH 6, pH 7, usually between pH 6 and pH 7. Many Pacific Northwest systems, both forest and grasslands, have fairly low pH levels, pH 4 to 6. And if you're at a pH 4, most of the phosphorus is kind of bound by iron. So the plants will not have access to any phosphorus that's there at that low of a pH. Once you get to pH 5, a lot of the phosphorus is bound to aluminum. And so you really have to work to increase the pH up to about 6 until you're able to release some of that phosphorus into the soil system and make it available to plants. So this means basically that low pH levels equals low phosphorus availability. And plants need phosphorus for ATP, for energy generation. They need it for a lot of their amino acids. There's a lot of different compounds where phosphorus is absolutely a necessity. It's one of the primary macronutrients needed by plants, right? So it's important to make sure that phosphorus is available, and a lot of our systems are phosphorus limited. So one way that has worked in many systems to increase phosphorus availability is to add lime, which is basically ground limestone rock. It's calcium carbonate and magnesium carbonate, and that works to increase the pH. And by increasing the pH, you're able to increase the phosphorus availability. So it's a way to do that without having to add a load of fertilizer to a site, by just adjusting the pH to better support the release of nutrients that are already there. Okay, I'm going to switch gears again to soil organisms, since we've talked about soil structure, soil nutrient cycling a little bit. And this is where I think it gets a lot more fun here with all the different organisms in the soil. So here are just some numbers. This again comes from the Natural Resources Conservation Service. Some just broad brush numbers on bacteria, on the microorganisms and some macroorganisms in soils, ranging from agricultural soils up to forest soils. And again, you can see with the bacteria, I think Gail mentioned some of these numbers at the beginning, at the low end, in 1 gram or 1 teaspoon, basically, of soil, there's 100 million to a billion bacteria in agricultural soils, up to, and that's maintains that abundance through prairie and forest soils. Just the composition, who's there, likely changes. With fungi, you can have several yards in agricultural soils of fungal hyphae, these long strands of kind of root-like structures. In forest soils, it could be several hundred yards. And even in coniferous forests, you can have up to 40 miles of microscopic hyphae in 1 teaspoon of soil. It's mind-blowing to consider that. And as we go down, I'm going to just focus on a couple I'm going to talk about more. Nematodes, these are also microbial organisms. Microfauna, there can be 10 to 20 in the agricultural soils, but several hundred in the forest soils. And arthropods, there's so many different kinds of arthropods, but there can be up to 100 in ag soils, couple hundred to thousands in prairies, and then tens of thousands in the forest soils. And earthworms, I know that varies site by site so much, but you can have as many as 5 to 30, and this is actually per square foot, 5 to 30 earthworms per square foot in ag soils, and up to about 50 in many deciduous forests. So really just mind-boggling numbers when you think about what's going on beneath our feet. in the soil system. So I'm going to walk through a couple of those types of organisms quickly here, just to give an overview. So this term photoautotrophic, this just basically means these organisms, these are bacteria that work kind of like plants. They capture light energy and use it to synthesize the carbohydrates or sugars using carbon dioxide as their carbon source. And one of the primary types of photoautotrophic bacteria that are important for soils are cyanobacteria, or also called blue-green algae. And these are a key component of biological soil crusts. Many of you have probably heard of biological soil crusts and seen them. They may not look like much to kind of an untrained eye or folks that are not familiar with them, but there's a lot going on in those crusts, and they provide a lot of really great services and benefits to the ecosystem. So they're common in systems with high light input that are much drier, usually deserts and grasslands. And they can, through their structural composition, they can help to aggregate soils, reducing erosion. And so in really windy areas, they're kind of help to hold the soil together. They can increase the soil surface temperature, which can be helpful for seed germination. Also, holding the soil together can be helpful for seed germination. And they modify soil water runoff by promoting infiltration through the topographic variation there. So having these little, miniature mountains of cyanobacteria helps to hold water in place, again, promoting seed germination for seeds that are there in those dry ecosystems. And they can increase soil fertility because a lot of cyanobacteria are nitrogen fixing organisms. So they can, again, take the nitrogen from the atmosphere and convert it into an available form of nitrogen that plants and other organisms can use. And you may have seen signs like these walking around Utah or parts of Arizona, where there's a lot of biological soil crust, and a lot of research and conservation efforts have occurred there. And the same is true in Eastern Washington, where in sagebrush and high desert ecosystems in Eastern Washington, there's a lot of really unique soil biological soil crust communities, stretching all the way to the Palouse. So definitely the conservation of the crusts that currently exist is really important. And also research has shown over the last about 10 to 15 years that it's possible to inoculate cyanobacteria into sites to kind of promote establishment of biological soil crusts that have been disturbed, and to kind of reintroduce biological soil crusts to kickstart their recovery. So that's another thing that can be considered for restoration of dryland ecosystems is reintroducing or inoculating the soil, biological soil crusts. Another type of bacteria that I think the only other type I'm going to focus on here is a big group of organisms called chemoheterotrophs. And these are organisms that are decomposers. They take some of that organic material and break it down into forms that plants can use. They include pathogens that can be very detrimental to plants. And they also include things like the symbiotic nitrogen fixing bacteria that I've already mentioned a few times, rhizobium, and then there's things called actinomycetes, Frankia and Parasponia species, all these different species of bacteria that live in and inhabit the roots and form these nodules on legumes, and are able to utilize the like I said, the nitrogen that's in the air and bring it into the plant and convert it into an available form of nitrogen that the plant can use, that is then cycled in the soil system. Okay, and then moving on to fungi, there are so many different types of fungi out there. We often think of things like these morels, right? When we think of fungi, we think of mushrooms. But there's lots of different types of fungi, and I'm really gonna just highlight a couple here as they relate to restoration. But they usually they all pretty much fit into three different categories: either decomposers, which are organisms that feed on non-living organic matter. What you see here in the kind of top right picture, this is a cut end of a log and the white rot fungi that's basically moving through the tree rings here and metabolizing the carbohydrates in this dead wood. There's also mutualists. These are organisms that provide mutual benefit to each other. And so this includes things like mycorrhizae. This is a picture of an ectomycorrhizal truffle that's, I don't remember if this is associated with an oak, but an ectomycorrhizal truffle from here in the Northwest. You can see these are some of the hyphae that are formed. And then also endophytes. So endophytic fungi can inhabit the roots and the stems and the leaves of plants. There are hundreds of different types of endophytic fungi. Many are beneficial to plants and there's a lot of research about endophytes in terms of how they can be used in agriculture to support plant growth and inhibit pathogens and attract pollinators. I mean, they do a lot of really cool things within the cells of plants. And this is just an image of several endophytic fungi that have been cultivated. And so not only are they functional, but they're also quite beautiful. And then finally, there's pathogens that can certainly cause disease or harm to their hosts. Often they're plant hosts. And you know, it's very common that there's interactions between these three types of organisms. So the endophytes could potentially help the plant to resist another fungal pathogen. So it's different fungi interacting with each other. Just quickly, there are lots of different types of mycorrhizae, mycorrhizal fungi. You all have probably heard of many of these. The arbuscular mycorrhizal fungi, which is represented here on the left, has what's called an arbuscule that basically forms within the root cell structure. And it's able to, the mycorrhizae itself is able to access additional nutrients and water and provide pathogen defense for the plant. And in exchange, the plant will feed that fungus carbohydrates that it acquires through photosynthesis. And the same kind of interaction or symbiosis is true for ectomycorrhizae. So ectomycorrhizal fungi generally form associations with woody species, so shrubs or trees. And it actually forms kind of a mantle or it forms along the outside of the root. And through what's called a Hartig net, it can exchange the nutrients and the carbon kind of by forming a structure on the outside of the root. So there's also ericoid mycorrhizae that associate with plants from the Ericaceae family specifically. There's mycorrhizae that form just with orchids. So as we learn more and we realize just there's how much more is out there. Seems like the more that's studied, the more is discovered, right? With this group of organisms. So this is something that we've been investigating on the prairies for several years, as have others in other parts of the country, and have found that especially in degraded sites, the introduction or reintroduction of mycorrhizal fungi can be really beneficial for native plant establishment. So we conducted a trial here in Western Washington prairies with mycorrhizal inoculation with fungi that were sourced from native plant roots and grown out over about a year and a half. And when we inoculated the native plants in the greenhouse and then planted them out in the prairie, we found that it led to a 13 to 300% increase in the establishment rates of 6 of our native plants. So this is something that we have incorporated into some of our practices with greenhouse production. and continue to be interested in terms of how we can utilize this in our restoration efforts. And this was work, and this just shows a little bit about why that might have happened. So Sasha Porter did her thesis on the same question and kind of expanded the species and some of the treatments. And she found here, you can see, this is with Micranthes integrifolia. On the left, these are when she was sampling for biomass, above ground and below ground biomass. You can see the roots that were inoculated with mycorrhizal fungi are quite a bit larger than those that were not. It's hard to imagine this little guy here in the control, the uninoculated, providing much for the plant in terms of the long-term establishment. So definitely you can see how it can benefit the below ground structure. All right, last thing, I just want to zip through a couple of the just incredible organisms that live in the soils that I think it's mostly just interesting to learn about, to think that these are, crawling around below our feet. First is nematodes. They basically fulfill every level of the food web. They're bacteriivores, they're fungivores, they're omnivores, they're herbivores, they're predators. And like they're actually the most abundant animal on Earth, with 4.4 times 10 to the 20 nematodes existing and inhabiting the surface soils around the world. There's a lot of research out there on nematodes. They're just fascinating creatures. And a lot of work is done to estimate their abundance, hence that very specific number. And a lot of modeling efforts on this. They also, I think, provide a lot of the inspiration for some really scary movie monsters out there. So I think they're just really unique creatures that fulfill a lot of different roles within the food web. Another kind of fun and interesting group of organisms in the Mesofauna category are Collembola and springtails. These live in the forest litter for the most part. They can reach densities of 200 to 800 individuals basically per cup of soil. They're really important for decomposition. So they are basically eating and metabolizing all that leaf litter. And they provide great food for some of the other macroinvertebrates like spiders and reptiles and frogs, or macroinvertebrates like spiders and other organisms like reptiles and frogs. So a lot of and huge diversity just in their structure and their communities. Earthworms, I think everyone here is familiar with. There's about 6,000 species that have been described. They feed on dead organic matter. They've been recognized as ecosystem engineers because they can really transform the soil structure. And they are really important for decomposition as well, because they can break down the soil organic matter. So generally, earthworms, if you see earthworms in your soil, that's a good sign that they're helping to decompose the material that's there. Ants also are considered ecosystem engineers. There's at least 20,000 species of ants that have been identified, and that number grows pretty regularly as people learn more and study more. They feed on all kinds of things in the soil system: ants, termites, little Collembola, mites. They are pollinators, they're seed dispersers, they protect seeds. Really fascinating animal or organisms with just a wide variety of functions within the soil ecosystem. And then finally, the megafauna. These are organisms that we can actually see without any kind of help, basically, except for some of those fossorial mammals that live in the soil all the time. So fossorial, meaning soil living species, also have great impact on the soil structure and functioning. The herbivores like pocket gophers, for those of you that are familiar with the Mazama pocket gopher here in Western Washington prairies, there's some voles and moles that really are important in terms of dictating where resources exist by their movement through the soil. And then omnivores and carnivores and ungulates, all that basically can dig and burrow and impact the soil structure, all have an important influence on soil resources. Okay, so last thing I'm gonna have here is just this image from a fairly recent paper that's just looking at soil networks. So, I showed you that early on, that picture of the soil food web, and there were a couple lines showing the connection between, connections between the different parts of the food web. Here's a much more complex look at it, where you're seeing the connectance between different organisms at areas that have recently been converted from farmland to a restored prairie, that it's already been 5 to 10 years, I think, so midterm through the restoration process, and long-term areas that have not been cultivated for 10 to 20 years and have had native plants, some native plant restoration. And you can see the different colored dots, or the different colored circles, represent different types of organisms: the bacteria, fungi, nematodes, earthworms, plants. You can see the bacteria and the fungi kind of dominate here. The size of the circle reflects the number of species within that functional group. The message here is that as you go through the restoration process, you start to see more and more relationships between the organisms. They're interacting more, they're providing more resources for each other. So you have this really complex web of organisms, of plants, of all of the different organisms I talked about are working together and interacting with each other to form this really complex and kind of jaw-dropping community that we can't see, but we know is there, maintaining the below ground ecosystem. So in closing, I would just encourage you to learn about your soils. You can learn about the soil type, the texture, the pH, your nitrogen and phosphorus availability. And depending on what you learned, it may be worth considering introducing or reintroducing some of the soil biota back into some heavily disturbed systems to help jumpstart the restoration process and support your restoration goals. And I'll say you can learn about your soils through working with your local NRCS office, your conservation district, or to sample and test your soils through a testing lab, or you can just send your samples away yourself to actually get your soils tested and get some answers to these questions. Okay, with that, I will wrap up and take any questions.
Okay, just a minute. I'm coming back in to monitor the questions. Sarah, thank you. That was so wonderful. That was great. We have some questions and I'll ask them in a moment. But first, as a segue to soil health, there's a wonderful webinar this Tuesday night, the 27th, and it is a Mountain Heather Ecology and Restoration Project and the Northern Cascades. So we can piggyback right on Sarah's awesome presentation of the soil and see what they're doing with the mountain heather. And then on the 29th, wildflowers, and it's an introduction to the flora of Washington and Idaho borders. So that promises to be truly lovely. So on to the questions and comments. Again, Sarah, that was just great. So a question from Michael is, does Sarah or others know of a similar resource on plant root depth / shape, similar to the Conserve Our Western Roots slide, but for Western Washington lowland native trees, shrubs, and ground covers?
Michael, that is a great question, and I have searched for that resource myself. I'm tempted to try to make one myself because I have not seen that. There is a great poster for Midwestern grassland species as well. And, oh, I don't think I have that. It's funny, I had that on an extra slide just in case before, but I think I took it out. So if you search for, Midwestern soil or Midwestern prairie root depth, that should come up. But I have not seen one for some of our lowland species. So yeah, that's something that I wanted to generate because I'm interested as well.
Okay. Next question is from James. Where does volcanic ash fall in the soil texture chart?
Oh, that's a really great question. My guess would be that ash would be, oh gosh. My first thought is that it would be on the sandy soil side, but it probably would have a bit of a sandy silty. It would be somewhere in the sandy silty mix because it's a larger particle. It's much larger than clay, right? So the particle size itself. So that's a great question, but my guess would be kind of like a sandy silt.
Okay. Another Michael asks, would wood chips be an okay replacement instead of sawdust to immobilize nitrogen?
Yes, wood chips can be used for basically the option of choosing sucrose or sugar or sawdust really depends on a couple things, but mainly the timeline that you want to immobilize that nitrogen. So the sucrose or the sugar is going to stimulate the bacterial community, which is going to act really fast, right? They're really small, they can turn over pretty quickly. So that's like days to weeks in terms of how quickly they will start taking up the nitrogen, but also how quickly they could then release it, because it only last a few weeks, because they'll start dying and releasing that nitrogen back out to the soil system. If you add sawdust, that's going to stimulate the fungi that are in the system a little bit more, and it's slower for the fungi to break down the larger carbon compounds that are in the sawdust, and even more so in the wood chips. So it will work, but it's going to be really slow acting, but it also will be longer lasting. So it will take longer for it to reduce the nitrogen content or availability in the soils, but it will last longer.
Okay.
Yeah.
Okay, and to piggyback on that, and this is my question, there is a lot of talk that I've heard among the master gardeners about biochar. And I don't know if there's been any work done on that to also cut down on the carbon.
Absolutely. I didn't bring up biochar here, but that is absolutely another option. So, and even just adding charcoal is an alternative to sugar or sawdust that has, almost permanent impacts on, the nitrogen. So it can bind certain compounds. It can bind inorganic nitrogen compounds to the structure, to the surface of the charcoal structure itself. It has huge surface area, all the little mini holes in the charcoal. Biochar also has the opportunity to be kind of inoculated too. You can use inoculated biochar, and it provides great homes, for microorganisms and some of those invertebrates. So it just helps with soil structure. It helps with immobilizing nutrients, but also can, depending on the type of biochar, can help to kind of maintain stable nutrient availability, and it can help to sequester carbon. So it definitely is a valuable tool in restoration.
Thank you. Thank you. Nathan asked, how did you source and scale the use of arbuscular mycos?
Yes, so the arbuscular mycorrhizae we sourced by actually sampling, taking samples of native plant roots in areas that did not have non-native plants nearby. And we worked with a mycologist down in Oregon to cultivate the fungi. So basically took the roots and the rhizosphere, which is the soil that's right around the roots, took that and basically spread it out and inoculated some what are called trap plants. So sterilized plants that grew in a grown in a greenhouse. And they grew those up and then took the roots and the soil from those and spread them out again. It's kind of like seed increase over about a year and a half by continually, harvesting and then re-inoculating and harvesting and re-inoculating. We were able to get a substantial amount of inoculum that we knew was just from the native plants that we collected.
Okay, and Jane asked, and this is so connected to what you just said, what is the preferred method to inoculate soils to regenerate it?
Yeah, well, that's great. A great question. And I didn't include this, but there is a really great resource. And maybe I can, if I can act quickly enough, I can put it into the comments. In fact, maybe, yeah, I can probably pull it up here. For the way that we did it is once we generated our native mycorrhizal inoculum, we mixed it in with the soil mix that we were using to grow the plants in the greenhouse. So that was actually when we were growing out plugs, we mixed it in, I think kind of the equivalent of 1 teaspoon per plug. And so that's one of the common ways to do it. There's been more research around just kind of using it, mixing it in with seeds or like creating seed coatings with inoculum. That's another way that, I mean, you'd have to kind of work with a group or, a company to do that. But it's also potentially possible to spread and mix into the soil surface. We didn't see as much success with that on the prairies, but that isn't to say that it couldn't be done. But there is a resource by Liz Koziol, Koziol, who has published a free kind of report on practical methods for soil inoculation. And she goes step by step on how to do that. And if you just look up mycorrhizal inoculation, Koziol, I think, Koziol, that should come up. I may be able to get it onto the chat as well.
Okay. Kimberly questions, do you recommend putting sawdust directly into the soil or composting it first?
Let's see. Oh, here's an example of the inoculation steps here. And the guide, the name of it is here on the side. Sorry, it's sideways. But if you want to look any of that up, that's something that you can find online. And I'm sorry, the question was to add the soil dust.
Should you recommending putting sawdust directly into the soil or composting it first?
Oh, good question. I would actually recommend putting it directly so you can kind of rake it in or turn it into the soil surface. If you add it into compost, you're also going to be adding the nutrients from the composted material. So that might, if your goal is to reduce available nutrients by adding sawdust, adding the sawdust alone is probably your best bet.
And those are all the questions. And I can't thank you enough. This was just wonderful. And I hope some of you will come back and join us tonight with Julie's talk on native pollinators that need native plants and how they help each other. So Sarah comments saying the talk was brilliant and it was wonderful. I'm not alone in my admiration. So thank you again so much for this. Enjoy
Thank
your
you all.
Good night.
And happy Native Plant Appreciation Month.
Happy Earth Day.
Happy Earth Day. Thank you, Gail.
Oh, you're so welcome.